But so for the people who are here right now, does anyone have any questions or any topics they specifically want to go over? I guess people will come in as we're going.
I guess one thing that I was reading over my notes from the unit last night, and one thing that was a little bit harder to understand looking back at it was the idea of kind of the water pressure, the Greek symbol of the water partial pressure stuff. Not entirely sure if a good umbrella term for that section of.
Yeah, the osmosis and the water regulation. Sure. Let's look for that. All right. Cool. So when we're talking, so for this quiz, I'm not going to ask you to do any of these calculations on these specifically. I just would like you to understand, you know, what forces govern movement in and out of a cell, right? So this figure is a little bit confusing because the way that the book presents it is a little off. They don't really explain where they get these values, right? So in these cases, you have two different things that they're looking at, two different forces that are creating pressure. So the first is this psi p, which is any forced pressure that you're putting on it. So that would be applying a force, right? Forcing the water to flow or salts to flow by actually applying force. So think of it as like a syringe or something. You're pushing on it, applying a force, and that's going to force something across, right? You can force something through a filter if you push it hard enough, you know, it might break though. What we're primarily concerned though is this psi s, which is the amount of salt, or generally we're going to be talking about salts, but it could be any type of other dissolved fluid, and that's going to innately create a pressure because of gradients, right? It's going to want to move from one place to another. One thing to note though is that cells are innately going to have their own type of internal pressure from water and salts, right? And so what you want to do is you want to look at what the base, you know, pressure is within the cell and then what it's being put into. And so when you look at this figure here, it's saying here that the salt concentration, water concentration of this right here is -7. So it's got a pressure, internal pressure of -7 pushing out. You then look at this environment. And so in this first case, we're going to put something into a hypertonic solution. So there's going to be more salt on the outside than on the inside. In this case, that's going to create a negative value. And since negative 0.7 is higher than -0.9, that means there's more pressure on the inside of the cell. And so water is going to be flowing outwards. And so one thing that I might have not mentioned to make it clear enough, but one of the reasons why the water is flowing in this case is because water is able to cross that membrane, right? And so this system could be put in equilibrium by the salt coming into the cell also, theoretically, but in most of these cases, we're talking about a cell membrane that doesn't allow those certain particles to come through. So salts and sugars, things like that. This is the water is what has to move. And so since there's a pressure difference, the water is going to move outwards. And so that cell is going to shrink. When you look at this second example, it's the exact opposite. So once again, we have this base cell that's at -0.7. But in this case here, we're going to be putting it into pure water, which has no salt. So there's not going to be any gradient. And so we're just looking at the internal pressure of that cell. And so in this case now, the pressure inside the cell, because, you know, there's salts, there's all these other things in the cytoplasm, is going to be negative 0.7, which is actually lower than the 0 of that neutral water. And so since now the pressure in the water is 0, which is higher, it's going to force the pressure in, right? It's going to force the water into the cell. And so that's going to be a hypotonic solution, right? And so what I want y'all to know about this kind of scenario is, you know, comparing high versus low, where water will move, but also that each of these, you know, solutes kind of has their own gradient, right? So if you are stabilized at, say, a concentration that's of salt water, say like a fish that's used to being in salt water, and you move that into an equally salty solution, but say it has like sugar or something in it, there's now a new gradient that's going to be created. And so that sugar is going to cause its own pressure. So the salt might be in equilibrium, but other solutes can be out of equilibrium. But yeah, mostly just where's the pressure higher, and then where is it going to force water? Because again, that membrane is going to be that semipermeable membrane is going to be blocking things that are not water from moving for the most part. Sweet. That answer your question or make sense?
Yeah.
Okay.
I think so. Thank you.
You're welcome. Let's see.
Wait, so when you're talking about like the cell and the solution, like, do you say hypotonic solution? Like, what would that mean? And then like hypo and hypertonic solution. Could you just go over that one more time?
Yeah, sure. So generally you're going to be usually you're talking about a cell that's being moved into a different scenario. So when you talk about hyper versus hypotonic, you're usually talking about the solution that it's being put in. So you're referencing that media that's changing. The easiest way to think of these, or at least for me to remember, is hypertonic means that there's too many solutes. Hypotonic means there's too much water, and I like to think of like a hippo. So hypo means there's too much water, so the cell is going to get bigger because that hypotonic solution is going to force water into it, and that's going to be where the cell bursts. Whereas hypertonic is hyper, too many salts, the water is going to be leaving the cell, and so that cell is going to shrivel up. Mm-hmm. Does that answer your question? Great. All right. Let's see. Anyone have any other questions? And obviously, oh, yeah.
Sorry, one of the questions on the study guide asked about why it's important for the cells in the xylem to be dead at maturity, but for the cells in the phloem to be alive. And so I was just wondering why those characteristics were important in order for these different types of tissues to operate.
Okay. One way to think about it is to think about what job each of these are trying to accomplish. And so when you look at the xylem, it's relying on a passive force, right? It's passively allowing water to travel up it by a mixture of capillary action and, you know, that transpiration, right? That loss of water is creating a water gradient and it's being pulled up. Think about if the xylem was full of cells that were alive, right? You would have barriers in between those cells. You would also have different things such as, you know, like nucleus and different type of organelles and things that are all located within that cell. And so that's going to make it harder for water to passively move through it. And so think about it as if, you know, that cell is alive, it's going to have innate force that's going to stop flow. It's going to have things in it that are going to keep the water from just directly moving, right? If you have organelles, if you have cytoplasm, that water is not going to be, you know, in as high of a gradient. And so it needs to be dead in order for it to have minimal resistance, for it to be passive. You know, as from looking at the phloem, you know that it is possible for obviously these living cells to pump nutrients and water, but that is going to use energy. And so the xylem is really good because it's a passive force and it's not going to require extra energy. It could be done alive, but it doesn't need to be to accomplish its job. If the phloem was dead, like the xylem, you wouldn't be able to create the two-directional flow. So I guess one thing I probably should have done to this figure here is put another arrow, because one thing that the phloem does is it does, primarily take sugars from the leaves and like those source cells and move them down to the bottom of the cell, but it can also move it around, right? So it can move nutrients back up. Say you have leaves that are being covered by another tree and they're not producing as much sugar, you may be able to have the phloem pump sugar to a different part of the cell or up or down. And so being alive allows it to do a two-way system and control the way that things are moving. But again, the xylem just doesn't need that to force that pressure to do it. Does that make sense?
Yes, thank you. And if it's okay, I also had one more question kind of related to that.
Yeah.
I was wondering, we discussed the type of transport, like apoplastic or symplastic, but I was wondering, because one of the questions on the study guide discussed what types of transport would occur in the xylem versus the phloem. And so I wasn't quite sure how to determine which would occur where.
Okay. So I think one thing that this figure, again, might miss out on is, if you remember, these are plant cells, right? So they have a cell membrane, but they also have a cell wall. And so when you have this like apoplastic root, it's not going through the cell membrane, but instead just riding along holes in that cell wall. And so that'll be like a difference between that, where it's going through there, whereas the symplastic root is going straight through the cytosol and all of that. And so when we go back to the xylem and the phloem, you can see that, since the xylem is dead, there's nothing inhibiting it. And so you have, primarily, I should put these closer, this symplastic root in which you have the water simply going through the center of these cells. There also wouldn't be any of this transmembrane root, because there isn't, that full cell wall or that cell membrane, because the cell is dead, right? So for the xylem, it's just going through the middle of these cells, which are now vacant. So you have symplastic root. And then for the phloem, it's primarily also going to be going through this symplastic root, but it's also able, depending on what needs to be pumped, this transmembrane route. So for instance, when you think of the phloem, there are these spaces, these walls where they allow two-way flow, and they have these capillaries and pores to allow stuff through. And so you're going to be pumping things through a membrane, but as well through these kind of barriers in between the cells. But I'll be honest right now, I'm not going to ask you that specifically on the quiz in terms of where, what two types of, or how the flow is occurring in those cases. I more want you to focus on the living versus dead and how that allows it to do its job. As a quick note of this one, like this apoplastic root, this is most important when we're talking about transport of nutrients from the roots. That's where you're going to see this type of movement the most, is water being absorbed, nutrients being absorbed through the roots, and then being passed through those roots to that vascular tissue within the cell.
Thank you.
You're welcome. Let's see. Any other questions so far?
Would we be able to talk a little bit about like the long night, short day plants and the short day or whatever the opposite is?
Yeah, sure. Oh wait, no. This is not the right PowerPoint, right? No. One second. Where is it? Okay. You can see this, right? It's pulled up. I'm sharing the right screen.
Yeah.
Okay, sweet. Where? Oh, okay. So, um, again, so this comes down to essentially yeah, the short day, long night idea is a little confusing. I feel like they could have made the terminology a little bit more clear cut. And so short day, long night plants are plants that would be starting to flower, closer to fall or winter, right? There are plants that require a shorter amount of daylight and a longer nighttime period in order to register um, germination. And that could be for a number of different reasons. It could be their way of like dispersal of seeds might require, specific conditions, or, perhaps they rely on animals in order to do their pollinating. So, the short night, long day plants might flower only during the sunlight. What I really want you to know is that essentially that plants are utilizing light in order to tell things about their surroundings, right? They don't have eyes. They don't have what we would consider the classic sensory systems that we have. So they're working in a different medium, right? So plants are using sunlight specifically and different wavelengths to tell them about the length of the day. And so in this picture, what we're trying to show is that light, what's happening in these cases is, the plant is being hit with a certain amount of light. And so this is something you'll talk about more in 108 in terms of how this gets transferred. But, the plants are absorbing X amount of sunlight. They have their specific, wavelength receptors that are picking up on specific things. And so that is being, say, this plant here is being hit by X amount of sunlight. When it gets hit by darkness, it's actually going to be doing a thing where it upregulates its genes. So it's going to say, all right, I'm in darkness. I've been in darkness for X amount of time. And now I can start producing hormones to start flowering, right? Because the cells need to give themselves a signal in order to start producing the appropriate materials to say, hey, bud, you should be growing, or hey, bud, you should be producing hormones in order to properly start creating your seeds or flower or anything like that. And so I'm not going to ask you again about short day versus long night specifically in their names. I'm more interested in knowing that like circadian rhythm is something that plants also have, and it's based off light. But that, you can interrupt plants' cycles by giving them different lights, right? By flashing a light on them, they now think that, daylight occurred X amount of time ago. And so the way that occurs is through receptors like this, right? They have different receptors that pick up on different waves of light. So for instance, during the daytime, you're going to be getting a lot more red light that's going to be occurring. You're going to have pure red light, yellow light that's coming from the sun. Once the sun starts to set, those rays, so this is an aspect of, of Earth geography or Earth shape, right? So you have the Earth, the sun starts to set, the wavelengths of the light that's going to be coming into our atmosphere are going to change based on the angle and how the intensity of the sun. And so when the sun starts to set, the light that is hitting us is going to be changing from that red to that longer wavelength, which is going to be far red. And so the plant can pick up on far red through that light and through nighttime through that wavelength. And so these proteins, just like if they were other surface proteins were to get ATP or come into contact with a proton or some other type of important material, it's going to change the shape. And so that changing of the shape is going to change how it reacts with other proteins. And so those plants that are, say, looking for a specific amount of daylight, they're looking for a specific proportion or a specific amount of time, depending on the species, in which this protein is either in, you know, if you're like a long day plant, you want to have a lot of PFR, right? You want to get a lot of red light. It'll turn this protein into this one. And then when you have enough of that PFR protein in your system, that's when your DNA starts going, okay, we have this PFR, let's start creating germination and hormones. And so it'll slowly build that up. Obviously, if you hit it with the far red light and start hitting you at nighttime, that protein's going to shift shape, and then no longer can that protein elicit the response of germination or hormone production. And so this is this is a specific color I'm talking about, but this kind of mechanism is how it works for plants along a spectrum of light. I kind of went on a long rant about that. I don't know if that answered your question or if that cleared anything up.
Yeah, thank you.
Okay. All right. Yeah. What I personally like is the fact that, you know, you can take, you know, this production, these seeds that are producing hormones, right? You can like cut off a part of another plant and attach it to a flowering plant. And those hormones will actually be forced and will cause that new plant that you grafted on to actually start growing. That's a cool thing that scientists have done in the past to try to figure out if things are hormone driven. Like they can, you know, take cells that are elicited and move them. So this class, we don't talk about like exoskeleton growth, like insect spider growth, but you can actually take the hormones or the cells from an insect that's undergoing molting, and you can inject it into another insect. And even if they're not ready nutritionally or time-wise or lifeline-wise, you can have them immediately start going through molting. And so that same thing works for plants, right? Where the sun is eliciting hormone responses, hormones are leading to changes in the plants. And, you know, as you change the amount of light, it'll change the amount of hormones that are being produced. I don't know for a fact, but I would assume, based on how other things generally work, that when you have, say, this, you know, this FR or this FR producing germination, it's happening constant. And it's only when it reaches a certain level or a certain amount of hormone present in the cell does it actually start germinating. And so, you know, these are passive things that are happening, right? The sun is hitting the plant, it's changing those proteins, and then those proteins are changing hormones. And so it's all happening without the plant thinking, just in response to the sunlight. Okay, so let's see. We have a question about plant pathogen response. Cool. So what I would like you to know about plant pathogen response is that it is far more generalized than our responses. So what I would like you to understand is that when a plant comes into contact with a theoretically toxic or pathogenic bacteria, fungi, or something, that plant is going to hopefully pick up on a specific part of that organism. So for instance, plants, you know, have these receptors on their cells like us. In this case here, we have bacteria. Not all bacteria are going to have the same receptors on the outside, but a lot of them, there are things that are evolutionarily held. And so most bacteria are going to have this beta glucan protein, surface protein. And so, you know, obviously, if it doesn't, there's a chance that the plant never responds to that pathogen and it might die. But as long as it has that specific reading, the plant will come into contact with it, and then it'll start producing the appropriate adaptive immune system response. And so it's not really an antibody, but it is specific proteins that help break down, you know, cell walls of bacteria, cell walls of fungi. But it's a very general response, right? So you can have a bacteria or a fungi that has evolved a resistance to this, right? Perhaps they don't have this on the outside of it, or perhaps they, you know, a plant's response is, oh, we're going to break down your cell wall with a chemical. Maybe that bacteria has a resistance to that particular material. And so the plant's response is going to be less energy intensive. It doesn't have to adapt to what it's doing, but it's going to be overall less effective because it's going to be less dynamic. And so it'll be a generic response for everything in that family, and it might not work. And so you have the immune response that I want you to know about, but I also want you to know about this hypersensitive response. And so, plants know, they've evolved, right? They've been evolving and dealing with pathogens for millions upon millions of years, billions of years. Fun fact, though, that I didn't realize until a while ago, but did you know that sharks actually evolved before trees? So they were swimming around in the oceans before trees started growing, So at first we just had, all those smaller plants, but they've been dealing with these pathogens and they haven't died out, right? And so the way they do that is they have multiple responses to deal with stuff. So not only are they picking up on that bacteria cell membrane, realizing what it is, producing a pathogen, but then what they're also trying to do is isolate that pathogen. And so the hypersensitive response is programmed cell death. As it finds the signal in the area, it's like, oh, there's bacteria here. That bacteria might be trying to spread. I'm going to close off all the borders to where I think that bacteria is. And so those cells, sorry, 1 second. The 3D printer started kicking off my material, and so I wanted to make sure it didn't break. So those cells are going to die around it, which is hopefully going to isolate the bacteria. Obviously, the bacteria might have made it out of that area. It might have taken a while for the plant to properly detect it, and so the plant's going to be using both of these responses. It's going to try to isolate the area, and then it's also going to try to use some type of generic response in order to kill it, in order to kill whatever the pathogen is. And then this is just, this is not something I need you to know specifically, but, as a cool response, certain plants can do even more dramatic things, such as, secreting these chemicals that actually make organisms smell to predators. Yeah. Okay. Let's see. So did that answer enough of the question about the plant pathogen response? Okay, great. All right, and then we have another question, which is, could you explain a little more about partial pressure and how it affects an organism's size? Sure. Let us grab. So talking about partial pressure, what I would like you to understand about that is this idea of surface to volume ratio and its relationship to partial pressure. So when I say that, what I mean is, partial pressure has to do with, I'm sorry. So I'm going to use both of these PowerPoints in order to discuss this. Let's see. All right, so partial pressure. So partial pressure is, this innate pressure that's being caused by, particles existing. And because of atmospheric pressure, gravity is always pulling everything in, right? And so when you have something like a gas, it's going to try to be pulled down. When you have a medium like water that's a little bit thicker, that's going to make it harder in order to keep those molecules within it, right? They're going to bounce out. There's going to need to be a larger amount of pressure to push that molecule into whatever you need to do, into an area. This has very big repercussions for how cells operate, right? If you say, take this water down here and you ignore that it's water and you instead replace that with a cell, this is going to be the same issue, right? You're going to have a certain amount of pressure that's trying to push oxygen, nitrogen, any type of important molecule into the cell, but only so much can go through it at a time. And so this is going to be a very large limiting factor for the size of organisms and how organisms distribute nutrients and materials that they need. And so organisms that are smaller are going to have a harder time keeping water and other materials inside of themselves, right? It's going to be easier to transport things. So partial pressure is going to force, oxygen into the cell, and then you can move that material, say this is oxygen, around the cell more easily. Or if you have two of these cells next to each other, the distance is quite small. So you can say transfer oxygen into the cell and then have that diffuse into the other cell if that cell has, a lower oxygen concentration. However, when you're talking about organism size, you have, say, a volume that is all their cells. And so small organisms are going to have a smaller amount of water compared to the outside of their body inside them. Think about this. If you were to say, take a glass of water and you were to put it outside in the sun, right? It would slowly start to heat up and it would slowly lose water and start to, it would slowly start to go away. If you were to say, take that water out of the cup and pour it onto the table, that water is gonna spread out. It's gonna have a much higher surface area and a much lower thickness, so a much lower volume. And so that water is going to start to heat up at a faster rate, and it's going to evaporate much faster. And so that is exactly what animals are dealing with. An elephant has a larger surplus of water in it. And so that elephant is going to lose proportionally less water, so they have to drink less often. Small organisms have to eat more often, and they have to drink water more often to keep themselves sustained. And so it's a trade-off, right? So they need to eat more, they need to drink more in order to sustain themselves, but their circulatory systems are a lot easier to manage, right? When you get small enough, you have insects. They don't even have a fully automated circulatory system, right? They just have a heart with an open casket, an open area that they're squeezing blood into. So when we talk about partial pressure and animal size, I'm interested in you knowing that it has both positive and negative consequences for animals of both large and small size. You have partial pressure, which is limiting the amount of oxygen that can be pushed into a cell or a set of cells. And so organisms, as they get bigger, need to have more complicated systems for getting that transported. But partial pressure also forces organisms to lose water and other important gases to their environment when they're too small. And obviously it's happening for the big creatures like the elephants and stuff like that, but it's just not happening at nearly as intense of a rate. Did that answer your question about the partial pressure, or do I need to say that more? All right, wonderful. Sweet. All right.
And does that have to do, I remember like one day in class, we were talking about like per gram of energy or heat, I think was lost, and like the difference between larger animals and smaller animals. Could we just like go over that a little bit more?
Yeah. So, yeah, this was at the end of class, so I had to like kind of gloss over it a little bit. So I might have not explained as much, but so we have two different graphs here, right? There's one on the left and one on the right. The one on the left is, it looks like it's completely linear, but that's actually a trick. This is a log scale, which means that it's actually dealing in more of a exponential fashion. And so obviously, you know, bigger organisms need more, need to use more energy, right? They just have more cells. It's obviously going to scale up. And so when you take this log scale and you put body mass by how much energy it's using, again, metabolism, our estimates for all of the heat being lost, all of the chemical properties that are being, chemical reactions that are undergoing, it goes, it increases, right? Elephants are using more energy than a shrew or a mouse. They just have to. But this graph on the right is this exact same one, except we're now taking that amount of metabolism that they have and we're dividing it by the size of that organism. And so if you look at this graph, it's no longer on a log scale, because the relationship is a lot more simple. If you look at just how much an organism weighs and then how much energy it's using, normalized by how big it is, you get this crazy graph. So like a shrew, though it's using a less amount of energy than an elephant, it is using proportionally way more than that elephant. That elephant is a lot more efficient in its metabolism. And so that is for a number of different reasons. But one of them being it loses less energy, right? It has a higher volume, and so it's retaining heat better. It's going to be retaining any type of nutrients or anything that might be passively lost, such as water, to its environment. And so that has a big effect on like how these different organisms expend their metabolism or their energy. So for instance, you know, looking at these shrews, right? We're looking at three different pregnant animals or brooding animals. You have a human versus a mouse. And so this mouse is going to be using, you know, most of its energy just to keep itself warm. It's going to be losing all that body heat because of that low surface to volume ratio, or volume to surface ratio, sorry. And so it's going to use some on reproduction and some on the metabolism and some on activity, but it's mostly keeping itself warm. As these organisms get bigger, again, the human is going to be using more energy overall, you know, 800,000 versus 4,000. It's a lot more, but again, proportionally, a lot less of it is thermal regulation. A lot of it is standard metabolism because there are more cells, right? You have more cells, so you have to have a base metabolism that is higher. You just need to, right? You need, each cell needs to undergo its same metabolic processes, so it's going to scale up. But everything else, such as thermal regulation, is going to be a little bit easier to do. So again, if you divide out how much this woman weighs by the amount of energy she's using, she's going to be using less than this mouse most of the time. And then this is also why scientists like to try to take, try to look at all of these systems in terms of like resting versus active rate, because, you know, when you're doing something, you're expending more energy, but we're trying to, you know, just look at baseline, right? What are your cells doing if you're not stressing them, if you're not doing any great activity, if you're not in this scenario where you're freezing and so you have to thermoregulate extra hard. All right. Did that make sense, or did that answer the question on the proportion of, you know, weight to metabolic rate?
Yeah, thank you.
You're welcome. Let's see. Can you explain number 3 on the study guide? Let me check. What was number 3 on the study guide? Can someone tell me? Let me pull it up.
It says, how do differences in blood pigment type show differences in an organism's environment?
Oh, thank you so much. All right. So So what I was kind of getting at with that question is to try to have you think of how organisms living in different environments are going to utilize different pigments because they have different levels of access to nutrients that they need. Specifically, when we talk about respiratory pigments, we're talking about the ability to carry oxygen, to your cells. And so when we're talking about these different respiratory pigments, and I'm not going to ask you the differences in names between them, what I really want you to know is that, we use hemoglobin that has a certain affinity for oxygen. Insects, which are requiring less oxygen overall, and they have, those trachea, they have those open areas in their body to let oxygen in, and they're thin enough that they're allowing oxygen to diffuse into their cells, they're going to be holding on to oxygen. They're going to need to do it less. And so they actually have a substance in their blood, which is actually made of copper instead of iron. And so it's less electronegative and it's going to hold on to oxygen less well. When we talk about seals and organisms that really, really need oxygen in their bodies, they have myoglobin. And so what's important about that is that myoglobin holds onto oxygen 8 times stronger. So that means that they can hold on to oxygen more. They can breathe in, their blood cells are going to take in that oxygen and they're going to hold it very, very strongly. The cells in our body that have myoglobin are our muscle cells. And so the reason that is occurring is because, our muscle cells are constantly doing work, right? We're moving, we're running. What happens when you run too much? You don't have enough oxygen in your muscles. And so your muscles start to produce lactic acid and then they start to burn, right? You start to get tired, your muscles start to ache. We have myoglobin in those muscles because those muscles are going to need to hold on to oxygen the longest. And so in our muscles, just like this seal, what's happening is the seal is diving, it's losing its base amount of oxygen. Once it gets down to its lowest amount of oxygen, then myoglobin is going to start losing that oxygen more readily. There's now a greater gradient because there's less oxygen in their body. They're in the water, so there's less oxygen around them. And so they're not going to be passively getting it as much. And so different respiratory pigments are evolved to exist in different microenvironments in order to hold on to oxygen at the level that these organisms need. Yeah. Does that answer that question enough? Does that make sense or? Yeah. Yes. All right. Wonderful. Let's see. Next question. Just to clarify, the larger surface to volume ratio means that the organisms lose heat more readily. Yeah, so you have to be careful when you're saying volume to surface ratio or surface area to volume ratio. So if you have a higher surface area, you have a lower volume ratio, and so the organism is going to lose more heat. If you have a larger volume to surface ratio, you have a larger amount of water in the middle, a smaller surface, and so you're not going to lose heat as quickly. So yeah, you got to think of, which one is larger and which one's on the outside. Let me know if that wasn't clear enough. All right. Next question. Also email, lab question. All right. Respond to that in a moment. Any other questions?
Can we talk about like open versus closed systems and the advantages or disadvantages to them?
Sure. Cool. So open versus closed systems are dictated by whether or not there's a separation between intercellular fluid and blood. So all organisms are going to have a type of blood that's going to carry things, but in an open system, all of the fluid that connects your cells and that blood are the same fluid. You have the same fluid flowing through your veins as you do that is inside your, that is surrounding your cells. Whereas if you think about it, I tried to make this analogy before in class, you know, we have blood in our veins, in our vessels, primarily kept there. If you cut yourself though, but you don't go deep enough to hit a vein or an artery, you're still going to have liquid that comes out of your skin, right? You're still going to have, you know, if you've ever seen like someone hurt themselves, like the meat of their muscle, there's still liquid in there, right? That is that intracellular fluid that is helping connect all of your tissues. And so in a closed system, blood and that fluid are separate. In an open system, they're the same. And so when you're looking at these systems, they each have their own advantages. So for instance, an open circulatory system takes less pressure to maintain. All of the pressure within your body is kind of being controlled by this heart pressure. By pumping your heart, you can maintain higher pressures. So one cool thing that I didn't get to talk about in this class, but I like to do in other ones is, you know, when you say, if anyone knows anything about fighter pilots, right? They have to wear special suits. When they fly at really high speeds, the blood pools in their legs. Why does that happen? Because you have pressure from the heart that's trying to push blood around, but the pressure of moving in the plane is so high that it actually forces the blood against your heart and it pools in your legs. For organisms such as, say, a spider or an insect with an open circulatory system, you would never have that problem. Why? Because let's say that the organism, the spider, needs more blood to be pumped into its head or its abdomen, right? It's just going to pump its heart a little harder, and that will cause pressure to go out through its entire body. And so that's what allows open circulatory systems to be used to do work or to have a secondary feature. So for instance, as I said, spiders pump their heart in order to extend their legs. So not only are they getting nutrients to their body, but by controlling, say, levers and muscles in their legs, they can pump their heart and just push out their legs. That's why when you squish a spider, its legs go from being out to curling in because you no longer have that blood pressure to help you. You know, when when a person dies, you know, their heart stops, they don't start to sag, right? Their body doesn't decompress because we're not using that pressure from our heart to control and sustain our body. And so open circulatory systems are better at just using that pressure for a secondary reason. And they use overall less energy. They're more energy efficient because, again, you don't have extra capillaries you're pushing things through. You don't have a separation of all these things. You're using pressure for secondary things. A closed circulatory system, though, is better because it allows you to control where blood is going better. You know, if you're too big, an open circulatory system is never going to work for you. You're never going to be able to get blood to those nitty-gritty areas in your body. On a larger scale, closed circulatory systems work better, but they also allow blood to be moved around to particular parts of our body better. And you essentially can, for instance, when you're eating, right? When you're eating and you need to digest, your stomach needs more blood. Your stomach is going to need more nutrients in order to process all your food. So what happens is, once you start eating and you start digesting, your body starts to regulate and say, okay, I'm going to pump more blood into my stomach and I'm going to use less blood in my extremities and other parts of your body. That's why you're feeling fine, you eat food, and then all of a sudden you're cold. You're cold because your body is now regulating where that blood is going to go. And so that's the advantage of a closed circulatory system. As, being able to sustain a larger, thickness of cells, grouping of cells, but also being able to decide where blood goes and assign that blood in a much better way, in a much easier method. efficient method. Sorry, is what I'm trying to say.
I have a question. So like when you get surgery, is that why, like, because this is kind of gross, but like I had to see the doctor and he showed me this video of this surgery I have to get, and I was wondering like why there wasn't blood everywhere. Like I thought there would be blood all like in the wrist, but there was like none. Is that why? Because it's separated from the other fluid?
Sorry, so let me cut out a little bit. So you're saying, you know, when you like look at surgery, like why you cut into someone and they're just not bleeding everywhere, right? Is that kind of what you're getting at? Yeah, exactly. So there is going to be some fluid, right? You know, when you cut into someone, there's going to be a little bit of fluid that comes out innately, and that's why I don't know if you watch the different videos, they're usually like, you know, they'll dab in areas, they'll try to make it dry, they'll put pumps in to kind of suck out extra of that fluid. You know, they'll kind of use a similar tube that's like, you know, when you go to the dentist and they put it in your mouth to like kind of keep it dry, right? That's exactly what's happening, right? If we had an open circulatory system and that doctor cut open into your chest to do whatever surgery, your heart pumping is just going to start shooting that blood out. It's going to be like if you hit an artery or a vein and it's some, you know, you ever seen Addams Family Values where they like do their skit and like they cut each other's arms off and blood is squirting everywhere. Like if we had an open circulatory system, that would happen during every surgery and we would have to find a new way to do stuff like that. But yeah, exactly. You know, the blood is primarily contained in those veins and arteries. So as long as the doctor isn't cutting into those, you're not going to be getting a massive pooling of it. You know, you're going to have blood from like the little capillaries and those small areas, but it's not going to be as dramatic.
Okay, thank you.
Yeah, you're welcome. Let's see. So we have another question, which I went over a little bit in the beginning, but it was on the concept question of comparing and contrasting human immune system and that of plants. So I didn't go too much into the human immune system, if at all. So really what I'm trying to get is like a basic concept of that comparison. And so, let's see. Boop, boop, boop. So I mentioned this earlier, but what I want you to know about plants versus human immune systems is that we have a very dynamic immune system that is constantly updating. It is finding new bacteria, it's finding new fungi, it is discovering what, you know, that pathogen is, and then it's attacking it, and then it's recreating an antibody specifically to destroy whatever that chemical is, right? We have a system that takes more energy because there's more steps to it, but it's also more dynamic. It's able to specifically kill anything that's coming into us. So that's, you know, that's exactly why the COVID vaccine works, right? They put a piece of the vaccine or the virus, you know, one of those receptors on the outside into our body. Our body picks up on it, says, oh, this is bad. I think I know how to kill this. I'm going to create some type of antibody to break down this cell wall or to destroy this receptor. Plants do not have that. The first time, you know, if you were to give a, you know, ridiculous, but if you were to give a plant a vaccine to the COVID virus, it would never get better, right? It would never respond differently. If it got better somehow, you know, it happened to kill that virus and you hit it again with that COVID, it wouldn't have an immune response to stop it from getting sick again. It would get sick again and it would try to fight it off. So what I want you to know is that plant immune systems are generalistic and try to respond to everything, so they take less energy, but it leaves them pitfalls which can make them get sick. That being said, they have a backup response that tries to help them, even though they're not going to generally be killing, you know, every bacteria or fungi, right? What the bacteria are doing is this hypersensitive response. Not only do they send out a specific task force, you know, chemicals to go kill that bacteria, they're then going to quarantine and try to isolate that bacteria from spreading to the rest of the plant. So they program cell death and they kill all the areas around that fungi or bacteria. Because, you know, if these cells are together, how does this pathogen move, right? Is it moving through the vascular system? Is it moving through cell walls to neighboring plants? The plant doesn't know. The plant doesn't care. It's going to try to just stop it the best it can. And so it's going to isolate that area, and then it's going to have a generic response. And so this is why, you know, we have issues where, you know, a single pathogen that's really good at contaminating a specific organism, such as, you know, the fungus that attacks ash trees. I can't remember what it's called, but, you know, those ash trees are not going to evolve to be able to kill that fungus because they don't have a response specifically for that fungus. The response that they have evolutionarily is not enough to stop that fungus. And so if it gets infected, it starts to die. And so that's why you can have entire species of plants that get wiped out by a pathogen. You know, a fun fact that you all might not know is if you ever taste like synthetic banana flavoring, like something that's like banana flavored, it doesn't taste anything like a banana you've probably eaten in your life. And why is that? That's partially because the bananas that we used to cultivate back in the day that we got that extract from have died out. They had a pathogen that came through and it killed all of our native bananas. And so all of the bananas that we're eating now are a completely different species than the ones we used to eat. And that's why they taste different. So this immune response can have a very dramatic effect on how plants are able to respond to their environment. Cool. All right, so then we have a question about number 13. So how does plant growth different from animal growth in life and mechanisms? So so what I would like you to know about that is two things. One, first and foremost, plants undergo indeterminate growth for the most part. So whereas our genes, our species, you know, different things determine how tall we're going to be, how long we're going to grow for, things of that nature. Plants don't really have a stop button. As long as plants are given the appropriate nutrients and time and sunlight, most plants can grow forever. They can grow until they die of old age, which might be a season, or if you're like a tree, you might be a great redwood where you live for hundreds of years without a problem. So the time scale that plants are growing are a lot longer. And again, they're growing just based on nutrients. You can take the same type of plant, put it in, you know, plant it in the spring, plant it in the summer. They'll have different amounts of conditions and times to grow, and that'll change how big they grow. The other thing I would like you to know about plant versus animal growth is simply that plants are undergoing a very specific type of growth. So plants are undergoing that secondary growth. Sorry, that secondary growth where you have cells expanding and pushing outwards. And then primary growth where again, you have cells that are growing in a specific regions. So part of what I want you to know is that plants are growing in specific areas. So, you know, behind this apical bud, or, you know, in these roots, in these lateral motions, whereas animals, let me open this. Whereas animals are growing in a very different way, right? We have a lot more types of tissues in our bodies, and those tissues are going to be replicating in different ways, right? Primarily about that, I would like you to know about, you know, just how plants grow. You know, we grow differently than plants, right? Our bodies get thicker, specific parts of our bodies start getting up and down regulated depending on what part of maturity we're in. But that's how much I would like you to know. We're not going to go into like cell cycles or anything like that. Just that, you know, plants are growing primarily and secondarily in those two ways, whereas animals are kind of always growing. I guess you can think of it, all animals grow in like a secondary fashion. They all have secondary growth, and they're always getting thicker and bigger in their way, and then their primary growth or height is limited and will only happen for a certain amount of time. Let's see. Then we have a clarification question, which is, what are the 3 different methods of transport in plant cells? Pretty much, are we talking about diffusion or transpiration and translocation? So when I'm asking that question, I'm talking about these different routes that can happen in terms of water going through a cell, through a plant cell. So, when you look at it here, plants have cell membranes that are covering them, but they also have cell walls. And so in plants, which doesn't happen with us because we don't have cell walls, plants have the ability of water and solutes to move through the cell wall without actually entering the center of the cell, without actually going into the cytosol, the cytoplasm, interacting with any of that. They can also obviously go through the cytoplasm. As I was saying, when talking about like the xylem, it's a hollow cell. So it's always going through this center part. It's always going through this symplastic route. And then that third method is the transmembrane route, which is a lot like the symplastic route, except when the material is moving between cells, instead of going through open channels, it's going to be moving over that cell membrane and that cell wall. So this could be because there's not enough space. Perhaps this opening in the symplastic route is not large enough for certain molecules to get through. Or maybe that molecule could never fully get into, actually, no, if it's going through the transmembrane route, it has to get into the cytosol. But yeah, so it just doesn't have the ability to go through there. It also could be something that the plant really needs. So perhaps, think about the phloem, right? There are those areas that have those little shivs, those little filters that go between cells. It's possible that in order to get a larger cell through, perhaps a larger sugar molecule, it needs to have an active transporter that helps it move across that cell wall. And so that's just what I would like you to know about those different types of transport. I would like you to know how transpiration transpiration causes water to be lost by plants, and how plants need to think about and need to have adaptations to keep water in them because of that process. But just in terms of like translocation, I mostly just want you to know about those different things, and that how some of it is actively pumped through, say, positive pressure or something like that. I'm not going to ask you too much about that, but I do want you to know like how transfer of sugars through the in the phylum are necessary, and kind of where they go and why the plant would want to move them. Yeah. Let's see. Let's see, can you go over question 22? Let me open that. All right, so 22 would be, how do red and far red light work together to aid plants? Oh, okay. So I talked about this a little before you got here. So let me pull this up. So what I would like you to know about this is that, plants obviously do not have eyes, right? They need to have a different sensory system in order to tell their environment. And so the way plants are doing this are, they're already taking in light for photosynthesis. They're already trying to be active when that's occurring. And so they've evolved multiple ways of interacting with that same light in order to tell things about the world around them. And so what I want you to know is that light, far red light and red light are what I used as an example of what is a multitude of different things, which are different types of photoreceptors, and the changes in these receptors changing when they're exposed to light. So what I want you to know is that, you know, plants, this allows plants to have circadian rhythm just like us. They get tired. They need to, you know, they're doing different processes based on whether or not they're being hit with red light versus far red light. One of the things that's important about that, though, is this is, you know, a passive process. So what's happening is they have these receptors, these PR protein receptors. They're making these, they're floating in their body. They then get hit with red light. The sun is out. It's primarily being hit with red light. That protein is then going to change shape with interactions with those photons. And now it's a different protein, the PFR. And so that protein now can interact with a multitude of other things in its body. So for instance, excuse me, you know, this PFR protein may be important for germination. So what's happening in the cell is it has this PR protein, it gets hit with light, it turns to PFR, and now that PFR protein is interacting with the DNA of the plant. And now it's saying, oh, we have PFR, we're gonna start producing some hormones. I know what this does. And if there's enough far red light, the hormones will build up in that plant and the plant will start to flower or start to fruit, or will start to, you know, open up its flowers or something like that. There's going to be some type of genetic or response to that plant. To that change in protein structure. So when we talk about far red and red light, I just want you to know that it allows plants to tell time, and it's part of a much larger set of of photomorphogenesis, or different ways plants have of interacting with light and telling things about their environment for them. You know, again, you can determine when plants are going to, you know, whether or not they're going to flower, depending on how long the days are. That is a function of that red light and that far red light, right? You know, if this plant is waiting until, you know, right before winter to spread its seeds out, it's not going to be able, it's going to need that light and that type of light, that proportion of light to tell what's happening. Because, you know, if it's going to be winter and you're going to spread your seeds out, they may just straight die if they're not protected for the winter. So it allows plants to make active decisions based off of passive forces that are happening to them. And, you know, this light also allows them to tell whether or not they're shaded, right? If they're not getting as much red light, perhaps they're underneath another plant or a tree or something like that. And so it allows them to, you know, to see essentially and respond accordingly. Sorry about that. Let's see. Cool. All right. Any more questions? Keep them coming. So we have a question, which is, let's see, is it true that animals with a higher metabolic rate will be more active than an animal with a lower metabolic rate? Not necessarily true, because there's a lot of different ways of measuring metabolic rate. And so if an animal is being active and moving around, it's going to have a higher metabolic rate. So definitely being more active increases your metabolism. But if you're trying to compare between animals, that's a little bit more of an iffy question, right? So let's go to here. So it depends on your scale, right? So when we're looking at body mass versus metabolic rate, as an organism gets bigger, it's going to have a naturally higher metabolic rate. Its cells all need to undergo chemical processes and things like that. So if you And so, each cell must be sustained. And so there's just no way that you're going to be able to, sustain an elephant-sized mass compared to a mouse with the same amount of energy. That being said, proportionally speaking, when you look at how much energy animals are using, if you are bigger, you're going to have a lower proportional metabolic rate. So when you say take that metabolic rate and divide by the size of the animal, it starts going down. And that's because, smaller animals are going to lose more heat. And so, an organism that's like, let's say we're going to talk about an ectothermic organism, one that is, losing or just has the same heat as its environment around it, it's not going to be talking about heat. And so its metabolism is probably going to be mostly higher if it's growing or if it's active. And so being more active will have it have a higher metabolic rate, but they're not necessarily tied to one another. Because again, if the mouse isn't being active, it still has a higher overall metabolic rate than the elephant, at least proportionally. So you have to be careful about what is being talked about in that question. Because again, like even on this one, I'm not going to ask you the differences between each of these metabolic rate, but these are 4 different ways of looking at the same system that I mentioned just very quickly. And there are even more types of metabolic rate that scientists may look at specifically at different things. And so this is one of those things that's very iffy depending on depending on what you're looking at. So you have to be very specific about your context. Oh, and my thing printed. Yay. So now I have these 2 pieces. I'm going to attach some spider silk to here. And then I'm going to move, it's going to move up and down on a load cell. And then I can see how strong that silk is. Yay. All right. Let's see. And then another question. Let's see. I mentioned in class that there will be fill in the blanks on the quiz. What will those look like? Is it just matching terms or definition? Will include more short answers. So in terms of the exam, there are multiple choice. There are some matching, such as, there's some matching, multiple choice. And then there are 2 different types of short answer. There's a few that are like 1-word responses. And then there are there's 1 or 2 that are like, think of like Mad Libs. It's like a paragraph where I've left out some of the words and I'm just asking you to fill in logically what that is. And so obviously for those questions are ones that I'm going to manually grade because I'm looking for specific answers, but there can be other answers in terms of our interpretation or where you go, especially in short answers. I like to, if there's a 2-parter, I like to base, if you're thinking about part B is right, but your answer to part A is wrong, usually I give you credit for part B, and part A, will be the wrong part as long as your understanding is clear. There will be 2 like essay questions on the test, but those are sentence long short answers as opposed to putting in a word. I just want you to give me, I think on each of them, I say how long they are, but like 2 sentences to answer one thing, 4 sentences, maybe 6 to answer another. As long as you feel comfortable that you've answered the question, that's fine. But there will be a little bit more short answer than in the past for that. And again, I like being able to read what you say and the context of what you're saying to kind of understand your thinking, to give partial credit and figure out where, I might have misrepresented something. So hopefully that helps. Let's see. Could you go over the difference between osmoconformers and osmoregulators? Okay, yes. So that is a great one because I would definitely like you to understand. Oh, this one, sorry. This idea. So, Wait, where? Oh, is it not in this one? Where is it? Homeogrowth and homeostasis. Is that it? Yes. All right. So, osmoregulation, and then let's see, do we still get an hour and a half to take the quiz? Yes. You got an hour and a half to take the quiz. You have 24 hours, so it opens at 10 a.m. tomorrow, and then it closes at 10 a.m. on Thursday. So if you really want, you could take it at 4 a.m. on Thursday if you're, feeling up for it. Sometimes I do my best work sleep deprived. I don't suggest that. But let's talk about osmoregulation. So osmoregulation is, trying to control how much water and salts are in your body. And so there are two different ways of dealing with this, just like dealing with temperature. You have organisms that keep their body the exact same water concentration and solute concentration as their outside environment. And you have organisms that can't do that because they're going to die. Their cells are not capable of living in those systems. And so osmoconformers like this octopus are just like those ectothermic animals. They're just sitting there in the water. And so they aren't going to, their bodies are just going to have that same, water solubility relative to their body. Osmoregulators, however, are ones that need to make sure that the salt concentrations are properly balanced out in their body. And so organisms still need, no matter if they're an osmoconformer or an osmoregulator, they're going to need to balance out those salts and protein and those salts and water in their cells, right? Because you can't help that diffusion is going to happen. Naturally, you're going to be losing water or salts to your environment when you pee, when you, I guess they're not sweating because they're in the water, but that's going to happen. And so osmoregulators have ways of pumping solutes in and out. And so osmoconformers usually don't worry about water concentration in their cells, essentially the cells constantly being bathed, so it doesn't have to worry about losing water, but they do have to worry about moving salts in and out of their bodies. So when you think of osmo, think more of water, so osmosis. So conformers have the same amount of water in their body as around them concentrations. They're playing with salt concentrations. Osmoregulators are also doing that, but they also need to care about the water that is inside their bodies. So they're going to have extra mechanisms in order to control that. And so when you think of an osmoconformer, they're all organisms that live in water. And then osmoregulators will be mostly vertebrates, but a lot of these higher up organisms, invertebrate versus vertebrate. The vertebrates need to control their water a little bit more. And so the way they do that is twofold. So, osmoregulation also has to occur on land. So organisms that are on land are still losing water, and obviously we need less. We need to take in more water than an animal that's in the water, right? Like we're losing it because we're not surrounded by it. A fish, however, is not going to lose as much water. And so think of terrestrial organisms as osmoregulators. A mouse is an osmoregulator. Certain sharks, osmoregulators. Osmoconformers, fish, octopus, things like that. In each case, they're both technically regulating something a little bit, but the degree to which they're doing it is much different. So, for instance, both of these organisms have to deal with water loss and consumption through eating. So, for instance, when a mouse eats, it likes to eat berries, it likes to eat seeds, things that have water trapped in them. So they get a decent amount of their water from eating, which is great because they're losing so much of it. Things in the water, however, will vary in their adaptation based on where they're from. So for a saltwater fish, so Nemo, it's naturally, as it's eating things, as it's sitting in the water, it's going to be gaining lots of water and salt into its body. So it's going to naturally, whatever its homeostasis is going to be thrown off by extra water and salt. So what is it going to do? It's naturally going to lose, water because there's so much salt in its environment, because it's being exposed to it. So what does it do? It excretes extra salt. So it's trying to change that concentration in its body. So it excretes extra salt. And then when it pees, and so one thing that's really important is like, how do different organisms pee? Well, a saltwater fish is going to be peeing out a lot of salt, and it's going to be having minimal amount of water. It wants to get rid of the salt and wants to keep that water in it. For a freshwater fish, it's going to have the opposite issue, right? As it's eating food, it's going to be taking in water. It's actually going to start losing salts in its body to the water around it. Its body is also going to absorb that extra water and start thickening up and start puffing, doing that hypotonic thing, getting fat. How is it going to control that? Well, it's going to be peeing lots and lots of water. It's going to be peeing out lots and lots of water. Imagine you've been drinking all day. All of a sudden your pee is very clear and white. That's because you're getting rid of all that extra water in your body. Alcohol is acting as a diuretic. You're peeing out extra water. That's what these freshwater fish are doing. They're getting rid of extra water to make sure that it's not too much inside of them. They also may take up salt ions in their gills. And so as an example of things on land, for instance, if you look at, like poop that's being produced by a mouse, if you've ever had like a rodent, what do? They poop constantly, and they also poop really, really tiny, dry, very, very dry poops. Why? They're trying not to lose water. So they're absorbing all the water out of their poop. And so when they excrete it, they're just excreting those base nutrients and minimal water. Spiders literally poop crystals. Spiders poop little white crystals. They suck out all of the water they can because they're even smaller. Organisms, again, that are in water, that are constantly getting things such as sharks and fish, they're going to be peeing a lot more water. Their poop is going to be a lot more liquidy. Think of a bird. Birds are eating fish, eating other small things, getting lots of water. Their poop is very, very liquidy. They're larger. They don't have to worry about losing as much water as larger organisms do. So I hope that answers the question. Let's see. Any more? Ooh, yes. Oh, okay, great. Yay. So how are we feeling about this unit and so far? How's everyone? We okay? We nervous?
I mean, a bit nervous because it's a bit nervous before every test, but feeling fairly confident at this point.
Okay. Yeah, I hate I hate giving tests, and I hate having to like watch people take tests because I feel really bad. I remember what it's like taking a test, and I just like feel bad. So unfortunately, generally, like if I was giving you a test, I usually bring candy and try to give it to people. Try to get some sugar / distraction from the pain we all have to go through. But I try to make it accessible. So yeah. But yeah, I'll definitely ask after the test if y'all thought it was fair, if how you felt that the study guide prepared you for the exam. So think about that as you're going through it, because I would really, obviously I want to make sure that it's an appropriate representation of what you've been taught and what I've told you to be, expected of you. So. Any other questions?
Could we talk a little bit about like double versus single circulation? And is that like similar or related to an open versus closed system?
Oh, are you talking about the different types of heart setups?
Yeah, yeah.
Okay. So I will tell you right now, there's not going to be any questions on like differences between the open or the closed circulatory system. So I'm not going to ask you any questions about comparing, these 2 versus 3 versus, one chambered hearts. What I want you to know about that is simply that there are a lot of different ways of doing this, and the circulatory system has gotten more advanced and a little bit more complicated as we've gotten more evolved, birds and mammals versus reptiles and amphibians. But what's most important about it, I think, is I want you to know that, in our system, in this closed circulatory system, blood is being sent to a secondary location in order to gather oxygen. In all of these cases, whether or not it's 2, 1, 3 lobe, 4 lobed, chambered, you're going to have this unoxygenated blood being pushed to either, the lungs of the organism, or amphibians, the skin, because they can breathe through their skin, which is so cool. And that is where it's picking up oxygen before it gets pumped to the rest of the body. Whereas when you're looking at these closed circulatory systems, the oxygen is entering through some type of pore. Let's see. Trachea. So for instance, these holes that are secretly on the surface of the insect is letting in air. That air is just traveling just like it would in our lungs to different parts of the organism. Since that organism is so small, that's enough to let the oxygen diffuse into its blood. Its entire system is this secondary area. Obviously, because this is blocking it. Sorry, it's a picture. Obviously, spiders and insects will have like their own versions of like lungs. Like spiders have book lungs, so they have these areas that also fill with blood. But they don't need that alone, right? They also have the secondary system that's doing it. So that's all I wanted you to know about the differences. I'm not going to ask about differences in heart evolution and stuff.
And I think we talked a little bit about like number 3, which is how do the differences in blood pigment type show differences in an organism's environment? And could we also talk about like the function of the blood pigments?
Yeah, so the blood pigments are there to make, to be very, very electronegative, to hold on to oxygen. The blood pigments are specifically there to make it, you can have oxygen that's sitting in your blood and being pumped around, and that will passively diffuse, but there's only so much oxygen that's naturally going to be pushed into your blood because of partial pressure and things like that. And so the blood pigments make it easier for oxygen to be held on, and so it'll be more efficient when transferring oxygen throughout your body. What I was saying earlier about the different types is that each of these like organisms, think about humans, seals, which are in water, and then insects. All 3 have primarily have a different type of this respiratory pigment in their body. And why is that? Is because they have access to differing amounts of oxygen, and they need differing amounts depending on what they're doing. So because insects have that open circulatory system, they're thin, they have air that's kind of just oxygen that's just pumping in and diffusing into their system. They don't need to have evolve as strong of a pigment to hold on. So they use copper in order to hold on to oxygen. We use iron. I cannot remember what the base of myoglobin is compared to hemoglobin, but myoglobin is stronger and able to hold on to more oxygen than hemoglobin. And so That's important for these organisms that are swimming underwater and will be without direct oxygen for a very long time. So seals need to swim around, they need to be able to hunt, they need to be able to hold their breath for a long time. So their myoglobin will hold on to oxygen and allow them to have a higher concentration of that in their blood, which then allows them to have it diffuse throughout their system over a longer time period. As I was saying before, myoglobin also exists in our muscles. So our muscles, as they're moving, are going to be using oxygen, and we can only pump so much blood to them. And so myoglobin lets them hold on to oxygen a little bit better. And so that's the one space you'll find it. But, you know, still it's not enough, right? You start building up lactic acid, your body starts to hurt. You know, if you thought doing like a thought experiment, if you were to say replace all of the hemoglobin in our body with myoglobin, what you would probably have is an inefficient system, because now you have something that's holding onto oxygen so strongly that it might not be able to diffuse into our cells. You know, if our oxygen concentrations aren't low enough to kind of force that, you know, the myoglobin in these animals are going to be happening once, you know, they're underwater, they're holding onto all this oxygen, they can't breathe, it'll slowly diffuse out. We are going to need it a lot more active. We're surrounded by oxygen. And so since we have this, you know, area where oxygen is diffusing, we're breathing, it's coming into ours, that myoglobin may never let go of the oxygen because it's never going to be forced to. There's not enough of a gradient. So I hope if that clears it up. Oh, so question that has not been answered, but what adaptations exist within plants to help regulate water? There we go. Where's the picture of the leaves? Oh, where is it? There we go. All right. So these type of adaptations come in two different ways. There are behavioral adaptations. So you have things like the CAM plants, right, which are going to be opening their stomata at a different time compared to plants that are in a more humid environment, right? They are changing when they're trying to undergo photosynthesis. So that's like one of them, this behavioral change. But really what I'm getting at the most is physical adaptation. So for instance, just like we have that epithelial tissue that covers our body, plants have mechanisms to keep water within themselves, right? So they have that dermal tissue with that cell wall, with that waxy layer on the outside to help keep water from exiting. So you have adaptations like that. You also have the stomata in general, right? Those open and close not only in response to nighttime versus daytime, but they respond to the amount of water in the plant and the amount of temperature outside. So if a plant has been going through a very dry season, it may not open its stomata very much. It's not going to get as much carbon dioxide, but it may not lose water and it may not dehydrate. And so the adaptations are that waxy layer, but also opening and closing of the stomata at the appropriate time, in order to regulate those in and out, those exchange of gases would be the two that happen the most. There are also adaptations that generally happen more specifically, or sorry, not generally, but specifically happen in different types of plants. So for instance, when certain plants' leaves, when it starts to get dry, they'll actually curl up on themselves. So the plant is no longer pumping water into a part of the plant that is not being beneficial to it. They will regulate where water is going. They may regulate where nutrients are, where plants in themselves may move in order to get themselves into a more favorable light versus not light spot. And then where is, and so, right, like one of those adaptations are in plants, right? So you have the stoma, which are allowing in carbon dioxide and trying to regulate how much water is being put into this area. You have the cuticle, but then you have these different layers and sheaths, which are, you know, even the ground tissue, which is being used for photosynthesis in leaves, is actively coated in a way to allow gas exchange to occur, right? They have the guard cells allow oxygen to move through the ground tissue, and that those ground tissue, whether or not it's being used for photosynthesis or for the Calvin cycle, will be separated or not separated. But the biggest things are stoma, behavior, and the cuticle. Still got some time. Any other questions? Also, for reference, we're starting to get to like what I find the most fun of like the course, just talking about like biomes, like adaptations. I realize in science, I just like weird environments and organisms that adapt to strange places. So like, you know, we're gonna start talking about like strange things, like, you know, if no one's gonna ask a question, like anyone ever heard of like brine pools, which are pretty cool? We'll talk about them tomorrow. They're like, think of a lake within a lake. So they're like concentrated areas of water that have a lot of salts, and that causes that water to sink in water. And so you can have like a denser lake inside of one. And that they're, you know, the first time scientists found a brine pool, they tried to like dive their submarine into it. And the water is so thick with the salts that it actually couldn't penetrate. It was too buoyant. It would keep bouncing out of the water. And then obviously it's so salty that like you'll like, I think there's a video I show is like, you know, a snake, an underwater snake, like goes into the brine pool and just dies immediately. Like the salt concentration is so high that it's toxic essentially to the organism. So we'll talk about cool environments on Earth. Oh, okay, let's see. Yeah, so someone's just asking a clarifying question of, you know, why might plants face an especially hard time with natural disasters and abiotic factors, you know, non-living things. And, you know, it's exactly what they said. You know, it's because plants are immobile for the most part, right? Their ability to respond to their environment is a very localized thing. And so they can't seek out favorable conditions like other organisms, right? Birds can migrate and fly places, and even small mammals or insects can fly or run somewhere. A plant, you know, most plants can't uproot themselves and move, right? And so all of these conditions that may be very localized are going to have to come with an innate response from the plant. The plant's going to need Yeah. All right. So, you know, for instance, you know, plants are, you know, in each of these cases, right? If an area suddenly has no water or it gets flooded or you salt the earth and you throw stuff on it, you know, that plant has to find a way to deal with that right now or it's going to die. And so it's going to have a lot more responses to something like this, whereas another organism could just be run away. But that also means that plants can only live in specific environments. That's kind of like what I was trying to get at last class in the beginning of this unit, was talking about the idea that, you know, plants, when they're, you know, going through an area, they, you know, if an area is always flooded, only certain plants are going to be able to live there. And so what plants are able to live in that environment or acclimate to the drought or the salt is going to determine what plants can grow, and then what other organisms are able to grow. You know, if you have a weed that is able to grow anywhere, but it has very low nutritional value, it might not be able to sustain any it might not be able to sustain plants, right? You may have an environment that, be able to sustain herbivores or insects, right? Maybe there won't be any insects in an area, but you'll still have weeds growing everywhere, you know? And so plants, you know, in their response to these factors are very limited, and will determine, you know, those factors determine what plants can live, and those plants will later determine, as we go up, you know, what other organisms can live in that environment. Obviously that angle is a little bit more about next unit, but, you know, I try to have everything should be connected, right? Bio should be all feeding off of each other. At least is the hope. Cool.
Thank you.
You're welcome.
Yes, thank you.
Yeah, take care.
Thank you.
Yeah, so we have, you know, 15 more minutes if any of you have any other questions.
Could you just quickly go over like I know there are a lot of different functions, but some of the functions of the ground tissue? So I know that the different types of tissue have more specific functions, and I know that the ground tissue can be used more universally for different things.
Yeah. No, where? Oh, there it is. I need to start naming things better. Yeah, so the ground tissue is similar to our like intracellular cells in the fact that they really, what they do is varied greatly depending on where they are, whether or not it's, in the roots versus it's in the leaves. So each, ground tissue system is going to be filled with the appropriate cells to do what needs to be done at that area, right? So ground tissue in leaves are going to be filled with chloroplasts. They're going to be situated and, made so there's a labyrinth in which air can diffuse through and can get to all of these chloroplasts and differing things. But then if you have ground tissue that is, say, located in, the roots, they may be a little bit more specialized in order to transport specific nutrients, right, across cell walls or to that vascular tissue. The ground tissue is what allows some of these roots to exist, right? Like going in between cell walls and things of that nature. In a lot of places, ground tissue acts like fat. It'll store sugars. But like, think of it as just the space where everything happens. The ground tissue is where you're going to be producing those antibodies. You're going to be producing pheromones. Ground tissue is going to be acting as storage for sugars, for water, and act as support. So it's a lot of just general, whatever you need. Really, I think when you're determining between the three tissue types, think of like dermal tissue is interacting with the world around you and keeping everything inside. Vascular tissue is keeping everyone healthy, getting all your cells water and nutrients. And then ground tissue is everything else that needs to be done to support those two systems. Yeah.
Thank you.
Yeah, you're welcome.
Thank you.
Yeah, you're welcome. All right. stop sharing.